Welcome to today's episode of the BioHub podcast. Joining us is Dr. Sam Clark, an accomplished physician scientist with a strong focus in the neuroscience space. Sam brings a wealth of expertise, holding both an MD and a PhD, and has dedicated his career to advancing our understanding of the brain and the nervous system. So thank you so much for joining us today, Sam.
Thanks for having me.
So perfect. Obviously wanted to jump in to discuss, your tenure at Terran Biosciences as CEO and founder. Some really exciting innovation and breakthroughs happening very recently. So obviously the approval of Cobenfy marks the first new antipsychotic mechanism in 70 years. Can you walk us through how this drug works and why this is such a pivotal moment for patients with schizophrenia?
Oh, absolutely. It's a very exciting moment. So up until now, all antipsychotics have really expanded on the mechanism of action of the first antipsychotic, which was chlorpromazine, thorazine. And that basically showed that by blocking the dopamine type 2 receptor, you could reduce hallucinations and what we call the positive symptoms of schizophrenia. But that can cause motor dysfunction, Parkinsonian symptoms, among other issues. And every antipsychotic that has come after that has had some interaction, whether full block or a partial agonist, but they mainly work through the dopamine type 2 receptor. There's other receptors that are thought to maybe play an additional role, but that's the main focus of antipsychotics up until now. Cobenfy is composed of two compounds, xanomeline, which acts by activating the muscarinic receptors, M1 and M4, two muscarinic receptors, and it's combined with trospium chloride, which blocks muscarinic receptors outside the brain. And the idea is that activating those receptors in the brain can be beneficial for patients with schizophrenia. But activating them outside, for example, in the gastrointestinal system, can cause side effects, nausea, vomiting, diarrhea. And so you need a second compound that doesn't cross the blood-brain barrier to cancel out the side effects outside the brain. So with this new mechanism, it does not cause the Parkinson-like symptoms that the other antipsychotics have been shown to cause. And so it offers a differentiated safety profile for patients, and it's also been shown in clinical studies to be highly effective as well.
Amazing. Perfect. So that leads on to talking about one of a really exciting drug in your pipeline, TerXT. This represents a breakthrough in therapeutic advancement. So can you explain what makes TerXT unique and its potential impact on the treatment landscape?
Sure. So we talked a little bit about Cobenfy, formerly known as CAR-XT, when it was under development. Now, one issue is that both of the molecules, xanomeline and trospium, are actually quite old. Trospium was invented in the 1960s, and xanomeline was invented in the 1990s. That's all about 30 years ago. And so early 90s. And so the thing is that those drugs are not necessarily optimized for the body, and they have to be dosed twice a day. So Cobenfy has to be taken twice a day, every day, and there's no long-acting injectable form. So historically, the way that antipsychotics have been turned into long-acting forms and this has been done many times before with other antipsychotics, are through the creation of what's called a prodrug. Now, what that means is you take the original antipsychotic molecule and you attach a little modification to the side of it, like a side chain. That side chain helps it get absorbed better into the body. And then the bodies in the blood, the natural enzymes in the blood, take that side chain off, which means now it returns to the original effective molecule. And so with those two things, that what we did is we said, okay, we will create the prodrugs of xanomeline and trospium. And we did, and we put them in animal models, and we showed that, for example, with trospium, we were able to improve it more than 10 times better absorption in those animal models than the original molecule. And that, we believe, will then enable, with the better absorption prodrugs, which TerXT is, the prodrugs of xanomeline and trospium, these prodrugs with better absorption should enable a once daily pill and a long-acting injectable form with a potential for a multi-month duration. And that would be a big breakthrough for patients with schizophrenia because medication compliance is an issue with patients, and having an
Mhm.
easier dosing regimen with right now with other antipsychotics, once daily is a gold standard, would help improve this drug for those patients.
Yeah, definitely. I mean, especially twice daily oral dosage is, even for most people, is sometimes you can easily forget it. So that would definitely be a massive challenge with Cobenfy. So I guess in terms of TerXT, how does TerXT improve the pharmacokinetics of the active ingredients xanomeline and trospium, and why is this innovation critical for patient outcomes?
Yes. So I think the way that it improves is by the addition of that chemical side chain. You form what's called an ester molecule.
Mm-hmm.
The way we did this is we had a team of up to 200 full-time chemists working on this to make well over 1,000 different versions. And of those, we refined to only the best ones that showed these big improvements. And by that addition together, that then was put into all the different screening studies to find the ones that would be the most effective. And then that, we believe, will then be a big breakthrough for patients by enabling the better, longer acting forms. So it's been a big project that's taken us about 2 years, and we're excited now to advance that forward. There's another reason it could be a big breakthrough for patients, and that is that the FDA frequently approves prodrug versions of drugs on an accelerated regulatory path called the 505(b)(2) pathway. And what that means is instead of having to do 2 big phase 3 studies, they often can do one pharmacokinetic bridging trial, which means you can get a drug approved referencing data from the previously approved Cobenfy, and usually in just about 5 years. And so that's a
Oh,
very
wow.
fast timeline for patients to get a newer version because you have a prodrug.
Okay, I see. So you've developed prodrugs of xanomeline and trospium, and the US Patent Office has given notice of allowance of that patent. So what were the key design principles behind creating prodrugs to enable once daily oral and injectable forms? And how does this approach benefit both patients, but also the broader medical community?
So, for example, what we had to do there was this initially was really difficult. When our chemists initially looked at it, they said, well, I don't think we can prodrug xanomeline. The way the nitrogen is, it would be very difficult. But
Mhm.
then through special technique called quaternization, as well as some other things, we were able to attach that side chain. And we then brought on a team of 8 of the world's top drug medicinal chemists. It's a special field of chemistry. And they worked around the clock to design over 10,000 different versions that then we worked with those 200 chemists to make over 1,000 of them and then test rapidly. So it was a big undertaking, but it should be beneficial not only to patients with these new forms, but also just in the broader medical community, because as all of our data publishes, you can see all the different medicinal chemistry we did for 2 years. And for example, a typical prodrug program may make 40 to 80 prodrugs. We made well over 1,000. So this was a very
Mhm.
large prodrug program
Yeah.
that used a lot of innovative chemistry that would then publish to show people, inspire other improvements to other molecules.
Wow, amazing. Um, Perfect. So not only Terran's collaboration has been with Columbia University, and this has led to the world's first software as medical device for the analysis of neuromelanin (MRI), which may aid in the diagnosis of neurological diseases such as Parkinson's disease, which is truly incredible. Could you elaborate on how biomarkers, particularly related to dopamine, are transforming diagnosis and treatments within neuroscience?
Yeah, so now we're shifting over from therapeutics to a
Hmm.
different topic altogether, the biomarkers. And that's something we actually did in collaboration, the biomarker part. With Columbia University years ago, there was a biomarker that's been shown to be useful for schizophrenia, Parkinson's, and other neurological conditions, a biomarker for dopamine. And dopamine is very important in those conditions.
Hmm.
It's called neuromelanin. And the way it works is all of our brains have dopamine, and every day a little bit breaks down and it binds to iron, forming a iron containing breakdown product called neuromelanin. And The thing is that that's detectable on any standard MRI machine. But up until now, there's been no way to really standardize and automate the measurement. So despite there being many studies showing its utility in Parkinson's, schizophrenia, no one's been able to automate that measurement and get it really accurate and FDA approved. And we work with Columbia University, where they invented some algorithms for this, and then we reprogrammed those, built it into a cloud-based software, and got it FDA approved. And this is the world's first FDA clearance for neuromelanin software as a medical device, and we think it really will help aid in the future for those doctors helping diagnose those conditions.
Yeah, absolutely. So out of interest, would that actually mean that they can be diagnosed slightly earlier than what's happening now?
It may lead to advancements like that in the future. There have been studies suggesting that. It will need more research, but so far, some clinical studies in humans have suggested that neuromelanin could help diagnose Parkinson's a decade earlier.
Oh, wow.
There has been research, for example, from Dave Sulzer out of Columbia, where he stated in his 2018 paper that he would recommend that people at risk of Parkinson's get a neuromelanin MRI up to every 10 years, as they get older, to help monitor for conditions. So it really is something that, with more research, could turn out to be a very valuable tool in that space.
Oh, wow, that's incredible. Fantastic. So how is Terran Biosciences' unique model of leveraging a small core group and assembling expert teams around scientific problems accelerated innovation and problem solving in what is a very, very competitive space?
So this goes back to when I founded the company. I said, look, we see all these tech companies moving so quickly to make things, and biotech moves very slowly. So I said, part of that is just the way the companies are laid out, very hierarchical, a lot of red tape. And so we said, look, what we're going to do is I built out a team of, highly trained core operators, and then we bring on teams of world experts specifically around solving specific problems. Like, for example, on these prodrugs, we rounded up 8 of the world's experts on prodrug chemistry and had them all sit down to solve the problem of how can we make prodrugs of xanomeline and trospium. And so the idea is that for this model, that's a model that keeps a lean core team and then very specific sub teams on very specific problems, as opposed to a more sprawling organization. That's enabled us to move very quickly to solve problems like these.
Amazing. Perfect. So one criticism of the pharmaceutical industry is a practice referred to as evergreening, where big pharma waits for their own patents to expire before advancing new versions of drugs. How does Terran's approach differ, and what challenges or opportunities does this create in the regulatory landscape?
Well, that's a good point you brought up. And there's been some criticism, for example, with certain other long-acting injectable drugs on the market, one of which, for example, was the 2-month long-acting injectable of Abilify, which was really only approved when the 1-month long-acting injectable basically went off patent. It was kind of timed to, and so the idea is that that is a criticism that has been leveraged at a big pharma industry, is that many times patients have to wait a long time to get the prodrug versions or the longer versions. And people say, well, it's patents, they're going to lose sales on the original version, so people wait. But look, at the end of the day, at Terran, we believe patients need the best version immediately. And so we said we're going to cut straight to the best possible version, which we believe to be the prodrug version. And that's why we said, let's make them right now. Typically you might have to wait to make prodrugs, but in this case, with xanomeline and trospium being old, they were ripe for prodrugging, and we say let's do it immediately. So there's a bit of a unique situation where a drug has been approved, and then right away there's these prodrug forms, close behind in development.
Yeah, that makes sense then. So obviously you have a unique model of drug development. So how do you prioritize which therapeutic areas or drug modifications to pursue, especially there are so many to choose from in such a vast field of neuroscience?
We're working on some new ones, and that the idea is that we look to see where the greatest unmet medical need is, where patients are suffering, and where something like a prodrug could make a big impact.
Oh,
that's really the
amazing.
main way we do it.
Yeah, probably the best way. Okay, and now obviously we've spoken a lot about Terran and the drugs that you're working on and the advanced medical, but just to dig into a little bit more about you, I know when we've spoken before, you've mentioned that your journey into neuroscience was deeply personal for you, driven by friends and family that have suffered with mental illnesses. How has this shaped the mission and values of Terran Biosciences with it being so close to you?
Yes. So when I was growing up, I was impacted by a number of friends and family members who suffered from severe mental and neurological disorders, especially bipolar disorder. And so I really was also frustrated by what I thought to be, when I was in medical school, the slow pace of drug development. So interested in neuro, my background, I went to MIT and I studied neuroscience. Then I went to Columbia University where I got my MD and my PhD, and the PhD was in neuro. And then I said, I want to go right into drug development where we can accelerate new treatments for people that are just suffering from these conditions, because I had just seen that the problems associated with these severe mental illnesses when there aren't great treatments available to people.
Yeah, absolutely. I think as well, when you've had a bit of a personal connection, it makes you work a little bit harder to try and solve the problems that are out there. So that totally makes sense. There's a lot of, you've mentioned throughout the podcast, there's a lot of exciting things coming up. So what excites you most about the future of neuroscience and Terran's role in pioneering new treatments? Are there any upcoming milestones or projects you're particularly eager to share?
So I think the biggest thing right now is just this breakthrough that we made with these prodrugs on top of the xanomeline and trospium. And we had only recently announced this, and there'll be more data publishing soon that we're really excited for people to see, just showing some of these massive improvements. After that, we do have some other exciting targets we're looking at, and that we feel we could also make a potential impact. And so, yeah, there's with milestones on this, we may look to partner this molecule, the TerXT, in order to leverage teams from even larger pharmaceutical companies to take it into the next stage. And so we're really excited to move forward on this and hope that it really benefits the patients who need these longer acting forms.
Yeah, absolutely. And it sounds like you're already doing such a great job, and it'll be very, very exciting to follow the journey of Terran over the next few years.
Thank you.
Well, thank you so much for joining us, Sam. It's been a pleasure speaking with you, and we look forward to hopefully having you on again in a couple of years with some even more exciting breakthroughs.
I really appreciate it. Thank you very much.
Thank you.